to deliver the welcome address for this 93rd International Virtual Learning Series in association with the Indian Pharmaceutical Association, South Tamil Nadu Local Branch, Head Office at Arulmigu Kalasalingam College of Pharmacy. Today we are proud to have an eminent personality, well-known teacher, undisputed researcher, and honorable resource person, Professor Dr. M. Saravanan, Sir, Faculty of Pharmacy, College of Health Sciences, Mekelle University, Ethiopia, to grace this occasion. It is a great pleasure for all of us that he could find time to be with us in spite of his busy schedule. Thank you, sir, for being a valuable part of this wonderful occasion. Sir, we are fully appreciate you for finding the time to be with us in spite of your busy schedule. Most welcome, Professor Dr. M. Saravanan, sir. Most welcome you, sir. So can you hear?
Yes, sir. Yes, sir. Audible, sir. Audible.
Yes.
Most welcome you, sir. Most welcome you, sir. Next, I hope this session will be a good platform for the students to get a good knowledge. My dear enthusiastic students will utilize this great opportunity to explore themselves. Finally, I welcome all the respected professor, faculty members, and my dear enthusiastic students for today's session. All the very best, my dear students. Once again, I welcome you all. Thank you.
And now it's my pleasure to introduce our speaker, Dr. M. Saravanan, sir. And shall I proceed, sir?
Yes, sir. Yes.
Sir, Dr. M. Saravanan, sir, was graduated in microbiology from Madurai Kamaraj University and doctorate with specialization in medical microbiology and nanomedicine from Satyabama University. Thereafter, he worked as a postdoctoral researcher through Israel government postdoctoral research fellowship in 2011 to 12 in Institute of Drug Research, Faculty of Medicine, the Hebrew University of Jerusalem, Israel, focusing his research on nanobiomaterials and their biomedical application. Prior to postdoc, he worked as an assistant professor, SRM University and Department of Biotechnology for 6 years from 2005 to 11. Presently, Dr. Saravanan is working as an associate professor under United Nations Development Program for the Department of Medical Microbiology and Immunology, Institute of Biomedical Sciences, School of Medicine, College of Health Sciences, Mekelle University, Ethiopia. His research specialization is development of novel biomaterials, emerging and re-emerging infectious disease and cancer. He has published more than 130 research papers in peer-reviewed journals, including high-impact journals, The Lancet and Nature, which is having an impact factor of 59 and with more than 5,500 citation and H index of 35 and I-10 index of 70. Dr. Saravanan is a collaborator in Global Burden of Disease study funded by NIH, led by Washington University. He has published 8 research papers in the Lancet and in Nature Medicine. He has participated in more than 75 national and international conferences, reviewers of more than 100 international peer-reviewed journals, and guest editors for various reputed PubMed and Scopus Index journals. Also, 3 edited book under process on nano strategies for combating antimicrobial resistance and cancer, TRC Press, handbook on nano biomaterials for therapeutic and diagnostic application, Elsevier Publisher, and handbook on cancer nanotherapeutic in Springer Publisher. He was received many international fellowship, grant, and awards, notably received an IET Nanobiotechnology Premium Award in 2019, International Fellowship for Advanced Course of Antibiotics sponsored by Institute of Pasteur and Foundation, Merits France 2016, and he has received International Union of Microbiological Societies Travel Grant in 2015 to IUMS Conference in Canada, and he has participated in pertussis, biology, epidemiology, and prevention meeting sponsored by World Health Organization in France, International Fellowship for 4th Advanced Course in Diagnostics sponsored by the London School of Hygiene and Tropical Medicine in UK 2013, Israel Government Postdoctoral Research Fellowship for the year 2011 to 12, and DST SERC Young Scientist Award in 2011. Received Best Researcher Award from the Chancellor of SRM University for research contribution and publication in the academic year 2009, 10, and 11. Received DST Young Scientist International Travel Fellowship in 2009 to research paper presentation in France, sponsored by DST Government of India, and research collaboration with Monash University Malaysian campus, doing collaborative funded project for the development of recombinant multi-epitope protein-based POCT diagnosis for multiple STDs in humans from 2017. Received TWAS and NORAD grant 2015 and 2017. And with these few words, I feel proud to invite our eminent speaker to deliver his address. Thank you, sir.
Hello. Listening my words, voice?
Yes, sir. Yes, sir. You are audible.
Thank you, sir. Thank you, sir, for very nice introductions. First of all, I would like to thanks to the management, Aralmigu Kalasalingam College of Pharmacy, inviting me for this international virtual conference, 93rd International Virtual Conference, association with Indian Pharmacist Association, South Tamil Nadu branch. I would like to thanks to the convener, organizing secretary, and all the team members. Those are arranged for this international webinar conference. So, first of all, I want to give you special thanks to the management, inviting management and the colleagues, the principal and organizing secretary, inviting me for these wonderful events. So I can go directly to my presentation now. My talk today about emerging therapeutic approaches for COVID-19. Before going to these presentations, I want to give you some information about COVID-19. So coronavirus, SARS-associated coronavirus 19 or coronavirus. This virus, we are struggling throughout the globe. We are struggling a lot during this pandemic time, last 6 months, more than 6 months, from the virus originated from China in 2019, end of 2019, December. It able to spread it throughout the globe within a short span of time. In the history of infectious diseases, the first virus is spread within a short span of time throughout the globe. More than 224 countries, it infected more than 15 million populations, so more than 15 million populations, and it killed more than 100,000 individuals, 1 million populations, around 1 million populations. Today we are struggling for the therapeutic options. There is a limited therapeutic supportive treatment option available for the treatment of COVID-19. I can discuss today about what are the emerging therapeutic options available or doing research throughout the globe. Different researcher in the different research institute struggling day and night wants to contribute for therapeutic option for the treatment of COVID-19 as well as the eradication of these pandemic situations. So these are the content of my presentations today. So overview of my presentations, including I have to give this overview of emerging and re-emerging infections and emerging infectious diseases and their global impact, and emerging therapeutic approaches to combat COVID-19. So in this, my main focus on these emerging therapeutic approaches for COVID-19, mainly for nanotherapeutic approaches and drug repurposing approaches, vaccine development approaches, convalescent plasma therapeutic approaches, antisense RNA technology therapeutic approaches, and traditional medicine approaches. And also I can give you the take-home messages, recommendation take-home messages after my presentations. So going to the presentations, it's infectious disease is a world of transitions. it's not a single country problem. The infectious diseases is not a single country problem. It's a global issue, global public health problems. Why? Because this microorganism easily transmit one country to another countries without any border, without any barriers, because it's an invisible creature. It easily transmit from one country to another countries. The some of the infectious diseases, the severity and magnitude, and the morbidity and mortality slowing down. Some of the infectious diseases, the severity and magnitude and morbidity, mortality growing up. What is the reason behind this? The main reason behind this, those are the diseases, the morbidity, mortality, and severity, magnitude slowing down. We have the appropriate treatment options or the rapid diagnostic method, as well as appropriate treatment option to eradicate this type of diseases. Particularly, we have the appropriate vaccine treatment options like poliomyelitis. We have the polio vaccines. Most of the countries throughout the globe is eradicated. 99% of the countries. Only some countries, African countries, there is incidence, but so most of the countries are polio-free countries because of the vaccine. Okay, this also this organism not frequently involving for emerging and re-emergence, but those are microorganisms. The morbidity, mortality growing up, the severity and magnitude increasing because of these organisms. It involving various molecular and cellular mechanisms. It able to resist the all the treatment options available in the market, as well as this organism, it emerging, re-emerging frequently. It's very difficult to produce the appropriate therapeutic option for the treatment of these microorganisms. So main particularly this antimicrobial resistance is a recent time, it's a very big problem throughout the globe is for antimicrobial resistance. Why? Because these organisms are emerging and re-emerging. It able to resist all the antibiotic available in the market. We can't able to treat with already available antibiotic. It's simply needed to for the development of new therapeutics, new antibiotic for the treatment of this antimicrobial resistant microorganism, including MRSA, VRSA, vancomycin-resistant enterococcus, ZSPL, CRE, and CRA. So I can my discussion today about only for COVID-19. This COVID-19 like virus, this it's a newly emerged virus in China, Wuhan. In the end of 2019, it able to spread it throughout the globe to 24 countries within a short span of time. Why these organisms? This some microorganism re-emerging because it involving for various molecular and cellular mechanisms. Some microorganisms newly emerged. Recent time. In 2000 to 2020, there is a 4 microorganism emerged. In 2002 is vancomycin resistant Staphylococcus aureus. And 2003, the same virus like SARS virus, so COVID-1, the coronavirus 1, it's emerged in China in 2003. It also spread throughout the globe, more than 32 countries within 6-month durations. And also other emerging viruses like Zika virus in Brazil, and also the re-emerging viruses like Ebola virus in Africa. There is many incidents throughout the globe. This emerging and re-emerging infectious diseases. That what's a pharmacist we should do for this? We can develop. It's urgently needed for the rapid development of novel therapeutic options, novel therapeutic treatment option for this type of emerging, re-emerging viruses is very important recent time. So my discussion today about the emerging, re-emerging infectious diseases, particularly the newly emerged virus, COVID-19. This virus, I want to give you this emerging and re-emerging infectious diseases. It's the emerging infectious diseases is a newly identified. It's a previously unknown infections that causes public health problem either locally or internationally. The recent emergence COVID-19 in a short it's an epidemic in China. After the epidemic, it spread because of the international travelers. Now the world is in fingertip. Maybe the travelers are traveling one country to another country within a short span of time. They may carry the virus to transmit the virus to the other countries. Because this virus, it's a unique virus, COVID-19. It's enveloped positive sense RNA virus. Most of it's a unique characteristics, not like other enveloped virus. Most of the enveloped viruses are heat sensitive and environmental sensitive. But this virus, this enveloped virus, newly emerged virus, is highly resistant to all the environmental changes, including temperature, all other pH, etc. Why? Because you have to think this virus, it is able to cause the morbidity, mortality, infection, and morbidity, mortality throughout the globe. It is able to cause more mortality in European countries as well as America. It's a very cold countries, as well as in so very temperate climate country, high temperature countries like India. That's why this virus highly resistant for all the environmental changes. And also it is able to spread the virus from one individual, infected individual, to another individual, infected individuals through airborne transmission, droplet airborne transmissions. This re-emerging infectious diseases is an infectious agent that have been known for some time, had reduced incidence to such low level that they were no longer considered public health problem, and are now showing upward incidence in prevalence in worldwide. For example, the recent re-emerging virus Ebola. It's re-emerged in 2015 and 2017, around 3 years. It is able to spread most of the some of the African countries. It killed more than 12,000 individuals in Africa. This is not a single country. I told you already, for the infectious diseases, the emerging and re-emerging infections, it's not a single country problem. It's a global problem. It easily transmits one country to another countries through the travelers or different type of the material we transported to one country to another countries. Normally we are dividing this infectious diseases into three types of infectious re-emerging diseases and deliberately emerging infectious diseases. The newly emerging infectious diseases, for example, COVID-19, it's a newly emerged and it's able to spread throughout the globe within a short span of time. The re-emerging infectious diseases, this infection is identified previously. Some duration is the incidence is disappear and re-emerging again and causing the infection. Some of the microorganisms re-emerge. It leads to inactivate the action of antibiotics, particularly antibiotic resistant microorganisms. Deliberately emerging microorganisms, particularly for this organism, most of the time they're using as a bioterrorism activities. Example like anthrax. Anthrax, it caused by Bacillus anthracis. It normally this bacteria, it able to produce endospore. It's possible to make into powder form, easily transmit into one country to another country through the parcel. Actually, I want to give you here. We should use our technology only for the constructive purposes, not for destructive. This deliberately emerging infections mainly for they're using bioterrorism activities. It able intentionally to killing for the animals or people. It's not good. That's why we should use our technology only for constructive purposes. What are the driving forces of emergence? The driving forces of emergence, the main driving force, it's an integrated driving force. It's the environment. This environment, the re-emerging microorganisms, because they're due to urbanizations and climatic changes. The microorganisms, it emerged or sometimes re-emerged, and also the pathogens also responsible for the emergence and re-emergence. The microorganism itself, it involving for genetic change or genetic exchange. It able to transmit the infection from one bacteria to another bacteria, one virus, one organism to another organism to become emergence, and also the antibiotic resistant organism itself is responsible for the emerging, re-emerging organisms and also cost. The cost also is for some of this human cost based upon the nutrition status of the coast, as well as travel history of the coast, and also population density, because crowded living conditions, it easily transmit the infection from one organism, easily transmit the infection from the infected individual to non-infected individuals, and also immunosuppression. Those are the patient or the human coast, immunosuppressed. Easily, they will get the infection from different groups of microorganisms because already the immunocompromised individuals are susceptible to all type of infections. So going to the COVID-19, I told you already, it's started from China and spread in Wuhan end of 2019. It spread throughout the globe. It causes millions of population, and it also killed around 1 million up to this most of the time. This first infection it caused in China, and it will transmit to European countries as well as America and also Asian countries and throughout the globe nowadays. This is the infectious disease expert meeting in every year conducted by WHO. The Director General of World Health Organization has warned the emerging infectious disease increasingly serious threat to global public health that require action across all over government sector and society. So this actually the history of World Health Organization. Two developing country leading for WHO. So mainly the WHO Director General, Dr. Tedros Adhanom Ghebreyesus from the working country Ethiopia, and also the former Deputy Director General and currently Chief Scientist, Dr. Soumya Swaminathan. She is from India, particularly in Tamil Nadu. These both people, they know very well about the developing countries' problems. That's why they're giving more emphasis on the eradication of the emerging and re-emerging infection in developing countries as well as developed nations. What is coronaviruses? The coronaviruses, it's a family of virus that include the common cold, SARS, and MERS. The most recent COVID-19 outbreaks involved a new strain that previously had not been identified in humans. That's why the name is called as newly emerging infectious diseases. The common sign and symptom of the coronavirus, it's already known. All the medias and all the media focusing on this. Is the main sign and symptom is cough, fever, chills, muscle pain, shortness of breath, or difficult breathing, sore throating, new loss of taste or smell. The lifespan of coronaviruses, many studies, many researchers, they've done the research throughout the globe. What is the lifespan of this coronavirus for different inanimate objects as well as living objects, including, for example, this coronavirus is able to stay in the plastic material 2 to 3 days, as well as air 3 hours, approximately 3 hours, and copper container 4 hours, and stainless steel container 2 to 3 days, and also all other material like cardboard 24 hours. Different studies, they proved lifespan of coronavirus in the different inanimate and living objects. The emergence of SARS-associated coronavirus, it's this SARS-associated coronavirus. I told you in 2003, the first SARS-associated coronavirus, SARS virus 1, it's emerged in China. It's the origin of this virus. Most of the time, the origin of this coronavirus, SARS-associated coronavirus, it's from bat. The intermediate coast, it varies from the emerging, re-emerging infections. That's the first coronavirus, SARS virus 1, that SARS virus. The one is the intermediate coast is civet cat. It acts as intermediate code. It transmits the virus to the humans. And also 2014, 13 and 14, there is this same virus is re-emerged and causing the infection in Middle East countries that we name is called as Middle East respiratory syndrome virus. It's the Middle East respiratory syndrome virus for the intermediate course of this Middle East MERS virus, MERS-CoV. It's camel. It from the camel because the eating meat and meat product and also living with camel. It is able to transmit the infection to humans. And also this recently newly emerged virus, it is able to transmit the intermediate coast till now unknown. They thought it pangolin is the intermediate coast of the newly emerged COVID-19. It is able to transmit it to humans. But the study is going on. What is that? How it transmitted through the pangolin to a human? Because we are not taking. There is many studies going on how which till now it's not identified which one is intermediate coast, but they might be thought pangolin is intermediate coast for the transmission of COVID-19. The structure of COVID-19. This virus, it consists of normally why we name is called as coronavirus. This spikes proteins look like cron. It consists of different type of structural compositions, virion compositions. One is spikes proteins. The spikes proteins, it's made up of crown. It's contain trimer structures, nucleoproteins. It's a nucleocapsid proteins and membrane proteins and enveloped small membrane proteins, and also these are the main structural composition of the virus. The virus consists of positive sense single-stranded RNA genome. It contains a different type of enzymes like protease enzymes, different type of protease enzymes, and also the genome is contained the sequence. Some of the gene of the genomes consist of RNA-dependent RNA polymerase and spikes protein genes, etc. This structural components is like it looked like the SARS-associated coronavirus. The replication of this virus, most of the time, the SARS-associated coronavirus, it is able to attach to the receptor site present in the human host. The most of the time, the receptor, the human host contain AC angiotensin converting enzyme receptor 2. It acts as a receptor for the attachment of the spikes protein of the coronavirus COVID-19. Once attached, it is able to involving entry inside the host cytoplasms and also release the positive sense RNA genome. The positive sense RNA is directly it involving for translation process to synthesize different type of viral proteins, and also it synthesizes that positive sense RNA genomes and assembly of the RNA genomes. After assembly, it involving for exocytosis mechanisms. The virus enter inside the host cell through endocytosis and through the receptor site after attachment to the receptor site, and also after the replication and assembly, it released to the exocytosis mechanisms. What are the factors associated with COVID-19 infections? There is the two major factors responsible for the COVID-19 infections. One is viral factors as well as host factor. The viral factor, we already studied about the structure of virus. The virus consists of different type of proteins, but mainly for spikes proteins, S proteins, the spikes proteins. The spikes proteins is able to attach to the host receptor ACU2, angiotensin converting enzyme 2, and these are normally spike protein contain S1 and S2 subunits. The S1 subunits determine the virus host range and cellular tropisms, and S2 subunits it's mediate the viral cell membrane fusions. And other protein like M proteins, membrane protein responsible for the transmembrane transport of nutrients that but release and the formation of envelope. And also other protein N and E protein severely accessory protein, and also it's going on some of the subunit of NE proteins. It's non-identified. It's going on research for identifying the unknown function of these proteins. And this coming to this Cost factor. The cost factor is highly responsible, particularly the COVID-19. It able to attach to the receptor. That name is called as ACU2 receptor. The host lung cells contain enormous amount of ACU2 receptor site. Once the virus enter inside, it through the mucous membrane, it able to multiply, attach to the ACU2 receptor and enter into the lungs. It multiply and circulate into the bloodstream. Normally, most of the time, the individual who are not more susceptible. So individuals who are more susceptible to severe diseases of the COVID-19 is elderly people, greater than 65 years old, people with underlying diseases. And after entering inside the human host, it attached to the receptor, ACE2 receptor present in the lungs, and enter inside the lungs, multiply to produce many number of prions. And also at the time, what will happen? Our immune system, it also the replicated virions circulate in the bloodstream. Our immune system is activated. It involving for production of cytokine. The main mechanism of the respiratory problem, respiratory storm, due to the cytokine storm, it leads to the patient not able to breathe. Finally, it leads to cause death, particularly for some underlying health condition, increasing the susceptibility of this virus, mainly for hypertension, chronic obstructive pulmonary diseases, diabetics, and cardiovascular diseases. Severe complications, those are having respiratory. Normally, most of the time, this virus is able to cause ARDS, acute respiratory distress syndrome, septic shock, metabolic acidosis, and coagulation and disinfection, and multiple organ failure. Sometimes it also leads to cause multiple organ failure. Very well, this virus able to attach to the receptor. This virus is origin from bat. There is some of the animal is animals act as the intermediate host. The intermediate host containing animal also should contain ACE2 receptor. For example, the bat, swine, and civet cases, it contain ACE2 receptor, but mouse not contain ACE2 receptor. For now, the difficulties the researcher facing, because most of the time we are using mouse as laboratory animals for most of the research studies, in vivo studies, but the mouse we can't use it for this research, COVID-19 research, because mouse does not having ACE2 receptor for the attachment of this virus. So we can go to COVID-19 infections from the entry to replication and immunoresponse. This virus entered through the nasal tract or oral respiratory tract or oral tract. Once enter inside, it through the mucosal membrane and reaches to the lungs. The lungs contain the ACE2 receptor. This virus attached to the ACE2 receptor, it involving replication cycle. After replication, it able to spread to all other parts of the human system, including the circulatory blood system. Once the circulatory blood system entering the virus, the blood cells is activated. It involving different type of mechanisms, immune mechanisms, mainly for the antigen presentations as well as cellular immunity and humoral immunity. The antigen presentation and cellular immunity, it able to produce various cytokine. Due to the high production of cytokine, the cytokine storm and it able to make for the damage of our lungs. The infected individual cannot able to breathe, as well as the difficulty breathing. They will get difficult breathing. Most of the time, it leads to cause death. The humoral immunity, it will takes a longer time, but those are recovered people. It produce humoral immunity. It produce the plasma cells and produce antibodies against the COVID-19. We are using for this convalescent plasma from those are recovered patient. We are using as a treatment option recent time. The pathogenesis of COVID-19, I already told you, this virus attached to the receptor, this ACU2 receptor enter inside, it involving multiplications in the lung cells and spread through the other parts of the system, including the blood circulatory system. Reaches to blood circulatory system, our blood cells, it produce cytokines against this virus. Cytokine storms occur, it leads to cause lung injury. Hello. Sorry for the interruptions. Hello. Okay. Sorry for the interruption. There is a power cut here. The generator is going on. Okay. So I'll continue my presentation now. Yeah. This the pathogenesis of COVID-19. It's already I explained. This coronavirus is able to attach to the contains ACU2. Mainly, this ACU2 is abundantly present in the lungs, and it reaches to the involving for endocytosis mechanism. It reaches to the lungs and multiply. The virus is able to multiply to produce progeny virion. The progeny virion circulate into bloodstream. Most of the time, the bloodstream normally once circulated into bloodstream, our blood cells it involving for production of cytokines, cellular immunity, production of cytokine. The cytokine storm it leads to cause lung injury. That's why we are using different type of supportive treatment for antioxidant. Those are the treatment we are using for anti. Hello. Hello, sir. Hello. Yeah. Okay, sir. Okay, sir. Actually, sir, I'm using my mobile network. Okay, anyway, it's a little bit slow. We can use it. The why sometimes that causes multi-organ failure for COVID-19 infection? So most of the people are these are questions because this microorganism, particularly this virus, able to attach to the receptor present in the host cell, particularly ACE2 receptor. The ACE2 receptor not only present, it's normally abundantly present in the lungs, but also present in other part of the human system, so mainly for kidneys, small intestines, as well as testis. That's why this circulatory, the progeny virion, it able to circulate into the bloodstream and further attach to other part of the body parts of the ACE2 receptor present in kidneys, small intestine and testis, it involving for sometime kidney failure. Okay, this and also multi-organ failure. The understanding why I have to give you this. The main focus, without understanding the cellular and molecular mechanism of COVID-19, it's very useful for the development of novel therapeutic options. We have to understand the molecular and cellular mechanism, then only possible to develop novel therapeutic options. This is a common drug discovery stages. If you want to discover the drug or the therapeutic options, we should select the target selections, lead discovery, very for novel therapeutic options for the eradications of COVID-19 and coming to the end of these pandemic situations throughout the globe. This different type of therapeutic options they following throughout the globe. One is emerging nanotherapeutic approaches, drug repurposing approaches, emerging vaccine development approaches, convalescent plasma-based therapeutic approaches, emerging antisense RNA technology approaches, and also traditional medicine. The emerging nanotherapeutic approaches to combat COVID-19, so mainly it involving various nanocarriers or nanobiomaterials. They're using it act as a carrier to carry the drug. Nowadays, we are using for COVID-19 treatment mainly for supportive treatment. There is no appropriate treatment option available for COVID-19. Only for we are using for different type of supportive treatment for the eradication of COVID-19 infections. There's many researchers, many nanotechnology-based research institute throughout the globe. They're using for nanocarrier approaches to deliver the supportive drug to deliver the supportive drug into the target site to destroy the COVID-19. So mainly they're using for liposome is act as a carrier, viral-like particles, as well as polymeric vehicle. This polymeric vehicle, it's one of the advantages, a biodegradable polymeric vehicle. It acts as a carrier to carry the drug and deliver the drug into the target site in optimum amount. It's a controlled drug delivery approaches. It's able to reduce the side effect, increase the biocompatibility of the drug, and destruct the targeted microorganisms, particularly COVID-19. This, if you want to go for the nano drug delivery or nano drug design for the COVID-19, we should consider these are the main important informations. We have to consider the route of administration of the particular nano drugs and drug properties. We have to study biocompatibility. It's very important, biocompatibility, because that's why most of the time the nano researcher they're using for biodegradable polymer. It acts as a carrier to deliver to carry the drug and deliver the drug into the particular site, and also in controlled manner. And also duration of delivery is very, very important. Nature of the delivery vehicle, ability to targeting, and mechanism of the drug release. The nano-based drug approaches is a main, for example, it's act as a nanoparticle plus the carrier. This the nanoparticle plus carrier. In case of potential viral, we have to use for this nano particle, the nano biomaterial. It acts as a carrier to carry the drug, deliver the drug. It able to inhibit the viral entry or also viral replications, blockage of viral replications, or blockage of viral receptor blockage. So mainly we are using for this nanomaterial encapsulated drug, deliver the drug to either to stop the entry of virus or to stop the replication of virus. Recently some researcher they used the particularly COVID-19. They're using for chemical moiety with the carrier And this chemical moiety with carrier is able to attach to the spikes protein of the COVID-19 virus. It is able to prevent the entry of the virus. It is able to prevent the attachment of this virus to ACE2 receptor present in the host human system. The another approaches like emerging nanotherapeutic approaches to deliver the drug. So mainly for these approaches they're using for nanoparticle loaded inside for target to lungs. This different type of polymeric material they're using to deliver the drug, as well as the liposome material and solid lipid material to deliver the drugs into the lung delivery, particularly lung system, deliver the lung delivery system. Mainly they're using as the nebulizer delivery. For example, this emerging nanotherapeutic approaches to deliver the supportive drug for COVID-19. Different type of supportive drug they recommended for the treatment of COVID-19, including different type of antiviral drugs, as well as hydroxychloroquines and also other drugs like dexamethasone, etc. This drugs possible to encapsulate or we have to encapsulate the drug into the carrier. We have to make in nebulizer form. This nanoparticulate, nanocomposite microparticulate drug, it is able to deliver the drug into the lungs and directly reaches to the target site and destruct the microorganism, particularly COVID-19. These are the main approaches the research is going on throughout the globe. The other approaches, drug repurposing approaches for COVID-19. These approaches they're using for different type of drugs, supportive drugs. There is no specific drug, new novel drug already produced for the treatment of the COVID-19. This many drugs, the supportive treatment option mainly they're using for the treatment of COVID-19. Why they're using supportive drug? Because already they checked for biocompatibility, safety and biocompatibility already studied. And also it passed for clinical trials 1 and 2 and 3 and approved by FDA. That's why most of the this recent time they're using to treatment of this COVID-19 patient for mild to severe infected patients by different type of supportive treatment options. So one of the main drugs, supportive drugs introduced in India, Glenmark recently, Flaviviravir. It's most of the time it's mild to moderate infection, COVID-19 infection. They're using for the treatment of this drug. The main the mode of action of this drug, it incompatibility of the receptor site to prevent the attachment of the virus into the human receptor. Involving ACE2 as well as protease receptor incompatibility as well as protease incompatibility to prevent the entry of virus, and also it involving for the prevention of the replication of this virus. And other supportive treatment including they're using for antiviral drugs like leponovir, remdesivir, remdesivir. Normally remdesivir most of the time it involving it able to inactivate the action of RNA-dependent RNA polymerase actions. And this one, leponovir and ritonavir, it involving to stop the transcription mechanism of the virus inside the host cells. The next one for dexamethasone. The dexamethasone is a common drug they're using for various purposes, and this also one of the drug for the commonly there is using commonly for nowadays for the recovery of the patient of COVID-19 patients. This drug mainly using to reduce the incidence of ARDS, acute respiratory distress syndrome. The next one for hydroxychloroquine and zinc. The hydroxychloroquine and zinc, the zinc plus hydroxychloroquine, it have higher efficacy and therapy, higher efficacy as well as compatibility. This zinc has been shown to show or stop viral replications. Hydroxychloroquines appear to allow zinc to enter the cell to disrupt the So very important approach. The recent time, many researchers throughout the globe are doing for research in emerging vaccine development approaches. emerging vaccine development approaches. This different type of vaccine development approaches, they follow many laboratories and group of scientists throughout the globe. So main things they following for 4 different approaches, including for the virus itself, either whole virus, naked virus, and sometimes they're using for recombinant viral proteins, recombinant viral vector, act as a vaccine for the induction of the immune response, and viral vector vaccines, protein-based vaccines, for it induce the immune response of the human host cells. There is a many researchers. There are more than 30 different types of vaccine throughout the globe. It's already coming under the clinical trial. Some of the vaccines enter into clinical trial 2 as well as 3. Some of them enter into clinical trial 1. This vaccine, the virus vaccine, the whole virus itself, they're using as a vaccine after the attenuation process. Attenuated viral vaccines. Viral vaccine, some researchers, around 5 different research groups, they're doing for attenuated viral vaccines. And viral vector vaccines, more than 5 different groups, they're doing for viral vector vaccines and nucleic acid-based vaccines, around 20 different groups. But many researchers, they focus on the vaccine development using protein-based vaccine, either spikes protein-based vaccine or membrane protein-based vaccines. This and other type of other approaches also, they follow in different researches. Many vaccines coming under the clinical trial, either nucleic acid-based or protein-based vaccine coming under the clinical trial 1 and 2. These are some of the vaccines already coming under the clinical trial phase 1, phase 2, as well as some of the vaccines already phase 3 clinical trials, including different vaccine like, these are the list of vaccines, recent time. It's already coming under the clinical trial 1 and 2, including for Oxford vaccines. There's a clinical trials going on in Tamil Nadu also. Some of the universities and hospitals that are doing clinical trials, SRM as well as other universities. Hospitals, they're doing clinical trials. The clinical trial and approvals. So actually this, if you want to get the clinical trial and approval for any drug or vaccine, it will take minimum 10 to 15 years. But the regulatory authorities like FDA now relaxed some due to the pandemic situations. They want to make this vaccine urgently. That's why they want to give some relaxation for the duration as well as some different things. This normally the COVID-19 other vaccine, it will take applied research and feasibility studies. It will take for around 1 year, and preclinical and technology development 2 to 4 years. Clinical development phase 1, it will take 1 to 2 years. Phase 2, it will take 2 years. And phase 3, it will 2 to 3 years. And also licensing, it will take 1.5 years. So altogether, it will take minimum 10 years for other vaccine or other drug developments. But the COVID-19 case, the regulatory authority is giving some relaxations and produce due to the urgent need of this vaccine for the eradication of this pandemic. And some of the vaccines already completed for clinical trial within 6 months, and phase 2, 6 months, and phase 3, 0 month. Maybe we hope to get this vaccine after 6 months. Most probably some of the companies, they may introduce this vaccine after getting the approval from FDA. The convalescent plasma-based therapeutics for COVID-19. The convalescent plasma comes from donated blood from the donor who have survived the virus. Those are recovered patient. We have to collect the blood from the recovered patients and separate the plasma. This plasma we are using as therapeutics. Mainly this plasma-based therapeutics not recommended by WHO and CDC, but only for the severely ill patient. Those are not able to recover the patient. They will go to severely ill, and last stage we have to try for this plasma-based therapeutic for recover for the COVID-19 patients. Next one, this approach also possible that some laboratories are focusing on for antisense RNA technology for the development of novel drug for the binding of the receptor to prevent the entry of the virus. It's possible, as well as they're doing for the translation of these particular viral proteins. Okay, they're using antisense RNA technology approaches to different type of researchers they're doing throughout the globe. And one more thing for the traditional medicine approaches, in particularly in India, the many governmental sector like Ayush, the Ministry of Ayush, recommending for different type of traditional medicines for the prevention strategies for the prevention of the virus. Most of the traditional medicines we have to use as prevention strategies. Sometimes maybe use as treatment strategies for like different antiviral traditional medicine approaches recommended by Ayush, Government of India. We are using for the treatment of this virus for the infected individual, and also we are getting good results. Recent time they giving the report, this traditional medicine giving good results. The main focus of this traditional medicines is either it able to prevent the entry of the virus or it able to prevent the replication of the virus. Most of the traditional medicine to eradicate the mechanism of the virus from the human host. These are the available supportive treatment for the treatment of COVID-19, mainly supportive treatments. There is this supportive treatment including for mild to severe and critical cases. Mild cases they're using for different type of antiviral drugs as well as chloroquine. Different type of antiviral drugs like heparin, remdesivir, and corticosteroids. Heparin and remdesivir and heparin and corticosteroids sometimes they used because there is a mechanism they identified from the postmortem report of COVID-19 patients. Some of the studies shows recently published in Nature, it involving for blood clotting. There is that's why we have to prevent the coagulation of blood they're using for heparin and corticosteroid, and also the yes. And those are many research laboratories. They're doing research in COVID-19. Not all the research laboratories, they're doing research because they need at least for biosafety level 3, so biosafety level 3 cabinet. Those are the laboratories, research institute, those are having the laboratory facilities for biosafety level 3, then we can carry on the research for the for efficacy studies, in vitro efficacy studies for COVID-19. These take-home messages for this presentation today, my presentations. The development of novel therapeutics for the treatment of COVID-19 is more urgent. Development of novel therapeutics for the treatment of COVID-19 is more urgent. We need to develop novel therapeutics for COVID-19 by working together. There is no action today, no cure forever. Yes, because the recent we are getting panic throughout the globe. All the human beings, human population get panic. Why? Because due to this pandemic, we can't be able to we can't be able to do our as usual work in outside, and also all the schools and colleges are closed for last 6-7 months throughout the globe. There is a lot of industries, lot of the productions, all the economic fall and most of the countries are slow due to this COVID-19. That's why this COVID-19 cases 80% mild and around 15% cases actually 80% cases are too severe and 1% cases were critical. Most probably it's around 1% to 2% cases are morbidity. It leads to cause the morbidity. Anyway, that we need to develop. It's very high novel therapeutic. Altogether, we have to develop, do the research to develop novel therapeutics for the treatment of COVID-19 is more urgent. There is no action today, no cure. These are my references. So we are also doing some, actually in our laboratory, in I am working in the medical microbiology department. We are also doing for the diagnosis of COVID-19. And with this also we are doing some research also in COVID-related research, including for some of the evidence already, 2 papers accepted and 1 already published, platelets report for lung inflammation for COVID-19 patients. These are the collaborated throughout the globe, including for Israel, as well as in India, as Malaysia, and other countries all over the globe, as well as I would like to thanks to the GBD projects. So next, these are the last year. Actually, I would like to thank WHO every year inviting me for some of the infectious disease expert meeting in sponsored by WHO Geneva. This is conducted in 2017 meetings. So any queries? Thank you. I would like to ask you any queries. If you need, please ask me now.
Thank you, sir. The forum is open for questions. Any questions? So thank you for your wonderful presentation, sir. So you told about the alternate therapies for traditional practices for COVID-19, sir. So, so now you have been in Ethiopia. So you know what are the traditional practices are going in India. So how the Ethiopia is different than traditional practices?
That actually in Ethiopia, like countries, is a lot of the traditional medicine available in Ethiopia, but unutilized traditional medicine. They're also using different type of traditional medicine, traditional plants. Mostly they're using different type of supportive treatment as well as traditional medicine. But not here, the government is not recommended for traditional medicines. But in our government, particularly Ayush, Ministry of Ayush, India for traditional medicine. Recently, I saw the newspaper. It's Kapusara Kudinir also recommended for the prevention strategies, not for the treatment purposes. It's most of the traditional medicines not approved. We can use, but because it does not having any side effects, we can use it as a prevention strategies. Prevention better than cure. That's why most of the time we are using different type of traditional medicine. Also, Government of India, Government of Tamil Nadu recommending various type of traditional medicine, including Kapusara Kudinir, I think, recently they recommended. Here also they're using different type of traditional medicine, but the government are not directly recommended. But they're using different type of supportive treatment, like recommended by CDC as well as WHO.
Okay, sir. So you have told you are involving in your lab is involving in the screening of COVID over in Ethiopia.
Yeah.
So have you found any pattern over the spread of diseases, sir?
Yeah, I want to give you here this very interesting pattern of spread of disease here. Here, like, particularly in Ethiopia, like African countries, it's a highly burden of diseases. But COVID-19, it's most 90% of cases are mild. If we'll give the positive, for example, we are collecting the sample, those are contact with the individual, those are having positive, they're also getting positive using RT-PCR method we are doing for diagnosis. Once we are getting positive, that patient most of the time he will say, I'm all right, there is no sign and symptoms. It's 90%, 92% of the cases positive carriers. It acts as a carrier. Normally, asymptomatic also is a problematic because they easily transmit the infection to susceptible individual, including they easily get the infections, and also it most of the time the elderly people, those are having other complications, they will die due to ARDS, acute respiratory distress syndrome. And here only 1% cases are critical. It's coming to critical. 0.5% cases showing mortality. So less mortality. Why? Because normally we analyzed why these developing countries less mortality compared to developed countries. Because the developed countries, they're following most of the time for hygienic procedures, each and every things. They're never get any other infection, including parasitic worm infections. So recent studies in Nature showed those are getting worm infection. It reduced the incidence of COVID-19 as well as these mild infections for COVID-19. Why? Because the worm infection is able to induce a Th2 helper cell activity. That's why most of the developing countries, particularly African countries, particularly Ethiopia, there's a less as well as mild infection in Ethiopia because of the people mostly is unhygienic. It's a good, it's able to get the commonly parasitic infection. The parasitic infection induce immune response. This immune response act against the COVID-19 infections. Most of the developing countries. This also on studies showed, proved, as well as published in Science recently.
Thank you, sir. Sir, what are the diagnostic methods you are involving in your lab, sir?
So we are using for RT-PCR, RT-qPCR, commonly we are using, and also the plant introduced for RT-LAMP method. It's only 60 minutes we have to get the result within 60 minutes. RT-LAMP method. There is no need for any sophisticated instruments. RT-LAMP method, we can see, visualize based upon the color differentiations. We are using some immunofluorescent tag primer. And we have to identify the positive COVID-19 positive samples with the NAC after the changes of color. This also planned to practice after some time, but it is commonly all over the globe. The WHO recommended recent time for RT-qPCR, real-time PCR. We are following the same thing, real-time PCR we are following to identify COVID-19 patients here.
Sir, what is your opinion on the second wave of COVID, sir?
I'm not getting you what you're saying. What is?
So there is a lot of, like you have mentioned a lot about Israel. So Israel, now there is a second wave of COVID-19 is going on.
Actually, there's some the developed countries as well as developing countries. There is a second wave, including for South Korea, as well as Israel and other countries, even China. Also, they're coming under second wave of infections. But there is no much information about second wave. The report of second wave. But if the second wave, maybe most probably the second wave of infection, it less virulence because this infection, it's one of the novel virus. It's enveloped virus. Most of the enveloped virus are highly sensitive, all other enveloped virus, but this virus is highly resistant to the environmental changes. That's one of the unique characteristics of this COVID-19 enveloped virus or COVID-2 virus. For example, once we infected to individual, it able to develop the herd immunity. The herd immunity, if you want to develop, it will take minimum for 1 year to 2 years throughout the globe. It needed more than 70% of infections, mild infection or asymptomatic infections. The second wave causing infection, most of the time, the virus is less virulence, not like highly virulence, the previous infection, maybe less virulence. Once the virus infected, it's mutated, most of the time it will reduce the virulence capacity. It also, one of the things, it reduced the pandemic within 1 or 2 years. The WHO estimated up to 2022, the pandemic will be over because of either the people will develop the herd immunity or the virus itself to reduce the virulence.
Thank you, sir. Any other questions?
So good afternoon, sir. This is Raghavendran from AKCP.
Good afternoon.
Sir, actually, during the starting stage of the COVID-19 in India, our Indian medical director, Dr. Ganesh, will be announced one issue, sir. Actually, the COVID-19 is not survived 15 days in human body. So if more than 15 days, it will be automatically destroyed. So people cannot panic and fear. So it's true or not, sir? why asking this question for you means because as a microbiologist, you have a more knowledge and details about the microorganisms. So
yeah, thank you so much for this very interesting question. Yes, it's correct actually. So the virus enter inside the human host. It reaches to the nasal or oral tract and also mucosal membrane. Normally, it reaches to the mucosal membrane. It able to multiply. It stay in the throat areas. It leads to throat infection 1 to 2 days. After it maybe multiplied, reaches to the lungs. Lungs contain the receptor, ACU2 receptor. The virus attached to the ACU2 receptor and enter inside the endocytosis mechanisms and multiply inside the lungs to produce many progeny virions. And once produced progeny virions, it circulates into the bloodstream. Normally, this lungs, normally 2 to 8 days. It stay in the lungs, it multiply to produce progeny virion. Once circulated into the bloodstream, what will happen? Our immune response, the blood cells, it act against the virus. It produce cytokines. That's why it leads to cytokine storm. The cytokines, the produced cytokines storm the lungs. The patient feeling they can't able to breathe and difficult to breathe in. It will take for 8 to 12 days. That after 8 to 12 days, maybe the severe cases will die within 21 days because there is no treatment or any complication. Elderly patients, they will die within 21 days. 21 days approximately. The non-symptomatic cases, what they will happen, it will not reach us. Maybe the viral number of virus enter inside the host cell also. It's very important on multiplications and also some of the immune mechanism. It able to eradicate the virus. It's well and good because this healthy individual, those are only immunocompromised and elderly patient are highly susceptible to get the infection. Most of the time it will go to critical conditions. Other healthy individual, it will not go to critical condition, but even though if you are taking for the support treatment, it will eradicate. It will be eradicate completely within 14 days. Yeah, there's the government of India giving the appropriate instruction because we should follow the government of health ministry instruction as well as CDC as well as WHO instructions. That's why most of the time they're keeping for quarantine. Those are infected people for 14 days maximum. It's maybe most of the time it will be the life cycle is over. Most of the time is eradicated for all the viruses due to the supportive treatment within 14 days.
Thank you, sir. I have one more question, sir. Recently, some the Siddha doctors can say the COVID-19 is not more complicated in chickenpox. So, what is your opinion, sir?
So, the COVID-19 not complicated like chickenpox, or?
Yeah, chickenpox. I mean, they will say the COVID-19 is very
clearly.
Yeah, I mean, the COVID-19 is very minor disease compared with chickenpox. So, what is your opinion for this statement?
Yeah, this is also because the chickenpox easily transmit and produce for different type of external sign and symptoms. It's look like it also easily spread. The chickenpox also easily spread, but it's not good because the chickenpox never causes mortality. The mortality very less chickenpox. What is COVID-19? The recent time in India is less mortality, I hope, and other countries are high mortality like European countries and America. I think it's not true. The COVID-19 is highly complicated. Chickenpox is only spread within the short community, so nearby communities. It will not able to go to other countries. Maybe it's easily eradicate because it's visible sign and symptom, but COVID-19, it's not showing visible sign and symptoms. Sometimes it's a mild, sometimes asymptomatic. The asymptomatic people easily carry the infection to transmit the infection to the susceptible individual. But the severity and magnitude, COVID-19 is, I hope COVID-19 is more higher than chickenpox. Chickenpox, it easily transmit. It able to transmit within the community, small areas, but easily identify then eradicate it. But COVID-19 not like that. It's very complicated to eradicate it. Maybe you have to think that we are facing the problem recent years, around 6-7 months. So the complete globes are closed, all the activities. So recently is now recovering, but even though recovering, for the infection rate is increasing day by day. It's very difficult situations. So I think the WHO also not giving the exact date when we go to the pandemic is over. They're saying approximately for 2022. Yeah.
Thank you, sir.
Thank you so much.
Thank you, sir. Any other questions? Any other questions? So what will be your final take-home message regarding this pandemic situation?
Yeah, the take-home messages for this messages is it's everyone, all the individual in the globe. It's not a single country problem. I said most of the emerging and re-emerging diseases. It's not a single country problem. It's a global issue. We identified the real part of this infectious diseases, how it spread within a short span of time throughout the globe, more than 224 countries within a short span of time. It's a global researcher, including the microbiologist, pharmacist. Pharmacist is a key role. You have to develop the novel, appropriate therapeutic option for this type of emerging and re-emerging infections. These take-home messages, we have to work together. For example, most laboratories, we should not have the state-of-the-art laboratory facilities, but we should collaborate with other research laboratories. We have to work together to contribute novel therapeutic option for these type of emerging and re-emerging infection. It will not, this won't be able to stop for this emergence. Maybe after 5 years or 10 years, there is new emerging viruses appear in the globe and also causes pandemic-like situation like this. That's why we have to work together, and also we have to work together with a collaborative approach, including medical microbiologists, pharmacists, medical doctors, clinicians, and also other researchers, biological researchers, and all the researchers work together. We have to develop the novel therapeutic option for the treatment of this type of emerging and re-emerging infections. Very vital, very urgent. There is no action today. There is no cure forever. This is my take-home message. You should take a message.
Thank you, sir.
Thank you, Dr. Professor Saravanan sir. We had a nice informative presentation, sir.
Thank you, sir.
On behalf of our management of AKCP, hereby I express my wholehearted thanks to you and your esteemed university for giving such an informative, wonderful presentation about the. Can you hear?
Yeah, yes, sir.
About the emerging therapeutic approaches for COVID-19. I hope this interaction will motivate and encourage them in active learning the therapeutic approaches for forthcoming days. So let's have a good collaboration in future also, sir.
Sure. You're welcome, sir.
Whenever, let me know, sir. Please let me know whenever would like to plan to Madurai. Definitely, we will invite our campus, sir, institution, sir. In future.
Thank you so much, sir. Thank you. Sure, I'll come. Thank you.
Another one final request, sir. Another one final request, sir.
Yeah, yes, sir.
Kindly let us know, sir, any of your friend colleagues, sir. Kindly refer your friend colleagues who are able to guide and support towards the development of our students' careers, sir, for our future webinar, international webinar.
Sure. I'll recommend through mails.
Please let me know, sir. Let me know, sir. Kindly support.
Sure.
Thank you, sir. Thanks for your wonderful support, sir. Thanks and gratitude for your wonderful support.
Thank you, sir. Thank you for your very good collaboration.
Let's have a good collaboration in future also.
Sure. Thank you, sir.
Thank you, sir. Take care, sir. Bye.
Bye. Take care, sir.